Well, dear students, today we will tell about new part of biochemistry, protein synthesis, storage and transfer of genetic information, or stream of information from storage of DNA to proteins. We will tell about central dogma of biology, so-called structure and function of nucleic acids, organization of genetic materials in cells, replication of DNA, methods of nucleic acid investigations, reactions of transfer of information, transcription, translation, for synthetic modification of proteins, reasons and consequences of changing of genetic information, called mutations, and the results of it, mutations, development of diseases, hereditary or maybe non-hereditary, and clinical significance of total each part of molecular biology. All of it in this course, but today we will tell about structure and function of nucleic acids, organization of genetic materials, replication of DNA, and the methods of investigations. And you know that main functional molecules in our living systems eat proteins. Without it, life in the form as we know it is impossible. And in our body work near 150,000 different proteins. All set of proteins called proteome. And each proteome coded by near up to 22,000 genes, so-called genome. And you can see proteome in any time more than genome. And because each gene can be source of information for more than one protein. Not every time it takes place, but very often. And in each cell, keep balance between synthesis of proteins and proteolytic digestion of proteins. Because we talked about it, lifetime of proteins in cells, it take place in region from any second to any month. And you can see a picture of different function of correspondent genes. Genes which will be coded any groups of proteins. For example, group of part of genome coded enzymes, for example, receptors and other intracellular regulative proteins. Intercellular connection, also intercellular and structural proteins, and a lot of ion channels and other multiple functions, multiple genes, but very big part. Now it near 40, but now it some less. But big part of genes, function of it products is unknown yet. Its number of gradually decrease, but not effectively discovered. And but we know total number of genes and different localization of each gene, because in the beginning of the century it was successfully resolved problem of determination of human genome. It was result of two discovered group, scientific group. It's international group which will be consists near 20 laboratories which actively provide it, and private laboratory, laboratory of crack vendor. No, and but now we have possibility to determine own genome if you can to buy activity of it, to buy this information from correspondent laboratory. And human genome consists near 3.2 billion nucleotides. You can to see it near 3.2, near the same. And you can to see that mammals, it's not too big genome, it's not maximal genome. Sometimes protists, for example, plants, amphibians, consist much more nucleotides, more than 100 billion nucleotides sometimes. But human genome, one of most complex, one of most multiple, and a lot of genes work in, participate in metabolism of brain. But again, you can to see comparison of size of genome. It you can to see very multiplicity of results, and all stream of information in our cells, really life, it's not stable structure, but it dynamic, constantly changing structure, and it really stream of information about protein synthesis from DNA to RNA, which described by so-called central dogma. It term was called by Francis Crick, really genial scientist, and it based on matrix synthesis, when storage of information in DNA and the realization as protein. Sometimes it's finished by RNA, for example, ribosomal RNA, and sometimes any other. And all genome associated in pieces of DNA, which call chromosomes. Chromosomes, it's 22 pairs of autosomes and 2 sex chromosomes, X and Y for male and X female, you know it very good, I hope. And one chromosome, it one very long DNA molecule, liner molecule with beginning and finishing, and size of it molecules from some tens millions nucleotides to some hundreds millions nucleotides, sometimes more, a little more. And the genes or pieces of genome which will be consist information about individual proteins distributed uneven, non-gradually, by islands, by parts, which will be distributed also not random, but it result of multiple very long evolution and recombination of different parts of genome from first living organisms to now to humans. And you can to see distribution of expressed parts of genome, different intensity of expression and frequency of expression. You can to see, and a lot of senseless information. But now we have information about it senseless, not random and have any significance. And you can to see average density in human genome of genes. It near 20 genes on 1 million base pairs, nucleotides. No, and again, I can to repeat, organization of human genome more complex than in other living species. And again, I wrote a little about history. First, describing about discreteness of inheritance properties. It was provided by Gregor Mendel in near 1.5 years ago, and evidence of chromosomes as carrier of information is provided by butterfly investigation by Thomas Morgan. Chromosome theory of inheritance. He was not only a biologist but also very effective morphologist. You can to describe chromosomes. And chromosomes and association with nucleic acids at first was opened and discovered by Friedrich Michel, which will be isolated nuclein as chemical substance and from white blood cells. But a very long time it was unknown significance of nuclein. Because it was known that it very long monotone repeating nucleotides, but is it have any significance? It's not interesting for scientists before beginning of previous century. But later, first evidence that it important, DNA important for providing of any functional significance. It was experiments of Frederick Griffith when transfer of properties of Rauth non-toxic colonies of pneumococcus to smooth toxic for mouse colonies pneumococcus, and it will be associated with transfer of DNA. And later it was effectively investigated by Oswald Avery, Colin MacLeod, and McLean McCarthy, which demonstrate that it really DNA, which it was winners of Nobel Prize respondent. And later it was evidence that DNA, it polymer with sequence of nucleotides which will be distributed as Chargaff rule when number of purine and pyrimidine bases, sum of it the same. And it, by results of Rosalind Franklin and Maurice Wilkins, which was described that it helical structure, and at last James Watson and Francis Crick describe mechanism of storage of information and transfer of information, again corresponding Nobel Prize. You can to see main participants in this stream of investigations, information about organization of life. And now it again describes central dogma of biology, DNA, which can be doubling by replication, transcription to synthesis of RNA, and translation to synthesis of protein, and alternative reverse stream of information from RNA to DNA. And not all DNA have information about protein structure, and it part of DNA, segment of DNA, with information about synthesis of functional active product, sometimes proteins, mainly proteins, but sometimes RNA, it referred as gene. And the main function of DNA is storage and transmission of biological information, but transmission possible by participation of correspondent enzymes. Well, about structure. DNA and RNA is polymers. Polymers of nucleotides. And the nucleotides, again, I must repeat it. But I see very often it information well known, but details of it information unknown for multiple students. And it polymers of nucleotides. Now you can to see what is a nucleotide. Nucleotide consists its stable structure, which consists nitrogen base, sugar, ribose or deoxyribose, and phosphate group. And maybe monophosphate, diphosphate, and triphosphate group with macroergic substances, by the way, especially diphosphate and triphosphates, but it also main participants of nucleic acids. And all of nucleotide bases divide on two groups, it pyrimidine derivatives and purine derivatives. Now we're going to see pyrimidine, it's single cycled heterocycle with two nitrogens. And purine bases, it derivates of purine. Purine is two cycled structure, heterocycle, with four nitrogens. And the combination of nitrogen-carbon, very effective for stabilization and chemical properties of its structures. And derivatives of pyrimidine, it uracil or thymine with double oxidative carbons, and cytosine with one oxygen and one amino group. And you know that thymine is a component of DNA, uracil is component of RNAs. And purine bases, it amino derivative adenine and amino and oxy derivative it guanine. And usually it finished by it information, but you can to see that possible two variants of distribution of resonance structures. It may be lactam form, lactim and double lactim form. More preferential equilibrium displays to lactam form, and it main form, but not avoided variant when will be lactim form, double lactim form, and correspondent structure will be other, and properties will be other. We will tell about it some later, but it real reason of mutation, it usual reason, spontaneous reason of mutations, a transformation to lactim form. And in this case, rule of Chargaff will be disturbed. And more than previous described it thymine or uracil and cytosine pyrimidine and adenine guanine purine bases. In nucleic acids present some other minor derivatives. It usually methylated forms. For example, methylcitidine, methyladenosine, guanosine, and methylguanosine, hydroxymethylcitidine, you can to see. and sometimes some unusual, for example, ribosylated forms, and or some non-usual interaction with ribose. Inosine, pseudouridine, inosine when absent of amino group, and also methylated form. Sometimes it can be sulfatated. It also can be present, and sometimes it special signals in DNA or properties of RNA. Pseudouridine or the hydroxyuridine derivates in transfer RNA, for example. And now with combination with deoxyribose or ribose and phosphate group, monophosphates, it adenosine monophosphate or deoxyadenosine monophosphate, correspondingly guanylyl monophosphate, deoxyguanylyl monophosphate, dimethylate derivates or uridylated form, and cytidine derivates, deoxy or ribo mononucleotides. Yes, and synthesis of it very important because it constantly must be metabolism of nucleic acids take place every time, and necessary to provide resynthesis of nucleotides, and it result of multiple transformations. Now you can to see, you know that ribose 5-phosphate it result of pentosophosphate pathway, when will be, by the way, reducing of NAD phosphate, which will be used for anabolic reactions and for keeping of reductive conditions, reducive conditions in cells. And the first transformation, it pyrophosphate association with pyrophosphate by ATP, phosphoribosyl pyrophosphate, active multi energy macroergic derivate, and later multiple amino acids will be participate in synthesis of, for example, purine nucleotides. For it, at first, it amino group as donor of glutamine, amino appearance of amino group, later interaction with glycine, forming of more long chain, later formylation, but donor of formyl groups, it again glycine. Glycine very important precursor of purine bases. Now you can to see two glycines will be participate in this. Later, again glutamine, appearance of amino group there. Later, forming of cyclic structure, imidazole group with two nitrogens. Later, carboxylation, association with aspartate, and aspartate will be transformed to fumarate, like in urea cycle, and derivate with succinate appears there, very similar with the urea cycle. Again, formylation, again precursor will be glycine, formyl group, and creation of cyclic form with forming of inosine monophosphate. You can to see multiplicity of its reactions. Now it's scale it's not too high, but you can to see derivates 2 glutamine and 1, 2, 3, 4 derivates of glycine and 1 aspartate, and a lot of ATP energy necessary for synthesis of inosine monophosphate, but later it will be transformed to adenine or amidation to guanine. You can to see there. It was purine basis for pyrimidine. Again, phosphoribosyl pyrophosphate. But phosphoribosyl pyrophosphate will be interact with so-called orotic acid, which will be synthesized by gradual transformation of carbon dioxide and phosphate groups, carbonyl phosphate, well known from pathway of urea cycle. But it not urea cycle, it carbonyl phosphate synthase 2. Carbonyl phosphate synthase 1 participate in urea cycle in matrix of mitochondrion. Carbonyl phosphate synthase 2 participate in synthesis carbonyl phosphate in pyrimidine base synthesis. Later, interaction with aspartate, very similar with urea cycle, but with some modifications. Synthesis of orotic acid, and will be associated with phosphoribosyl pyrophosphate and synthesis of uridine monophosphate. And later, methylation lead to thymine, or maybe amination will be lead to cytosine. It's about synthesis. And about structure. No, you know that association of nucleotides one to other, well known from NAD and FAD. It's association by phosphate groups. Nucleotide in NAD, nucleotides interact by phosphate groups, by tails, and nicotinamide adenine dinucleotide and flavin adenine dinucleotide. But in DNA and RNA, association phosphate group interact with two riboses or deoxyriboses. Now it DNA deoxyriboses, RNA it riboses, and it phosphate group associated with third carbon of one ribose or deoxyribose and fifth carbon of other ribose and deoxyriboses. And this structure non-cyclic, it linear structure, and if we'll be take any chromosome, it some millions, some tens or hundreds millions nucleotides, but it linear structure, and move it along chromosomes. At last, will be paired to phosphate group, which will be associated with 5' 5 carbons of ribose or deoxyribose, and non-associated with next ribose or deoxyribose. Phosphate group will be free. It will be called 5' end. On the other side, will be moved, and will be achieved at last third carbon hydroxy group, non-associated with next phosphate group. It's 3' end. And direction, again, phosphate, free phosphate on 5' end, and 3 phosphate, fifth phosphate on 5' end, and 3 phosphate, 3 OH group on 3' end. And reading of it sequence every time beginning from 5' end. Now it for DNA ATG, not GTA, ATG. For RNA, now in this sample, UGC, uridyl guanylyl cytosine, not alternative. Every time beginning from 5' end and finishing of 3'. And DNA is double strand of linear structures, and it will be consist complementary nitrogen bases associated one to other, and every time now it's possible to remind Chargaff rule. Cytosine every time associated with cytosine to guanine, adenine with thymine. and by weak hydrogen bonds. And orientation of strands alternative. On one side 5' and on one side of one strand directed to 3' and on other side of this linear structure. But second strand orientate alternatively. When was 3' end, it was 5' end of second strand, and finished 3' end or 5' end. And hydrogen bonds, which will be create between nitrogen bases, it's very weak. You know that it's compatible energy with energy of thermal chaotic movement. But number of wheat bonds a lot, multiple of wheat bonds lead to stabilization of its structure. And additional stabilizational factor is so-called stacking interactions between hydrogen bonds. Really, other part of nitrogen bases is hydrophobic, and it leads to additional stabilization, regulation of expression of corresponding genetic information from DNA. And it's multiple of its parts in DNA, and especially important for RNAs. For RNAs, sometimes may be very complex structure. You can to see stem loop variants of folding, and sometimes you can to see it very complex interactions with forming of specific conformational elements which important for functions. Now you can to see ribosomal example of ribosomal RNA with multiple stem and loops configurations with possibility of changing of its structure, and all of it signals important for functions, signals for interaction with corresponding enzymes. Well, and now about now you can to see comparison of size of DNA. Really, it's very long system with comparison with protein structure, diameter of DNA compatible, not some less than size of corresponding proteins. Hemoglobin is not too big protein, but really working with DNA and RNA provided by proteins sometimes much more molecular weight than hemoglobin. Comparison with water molecule, amino acids, and compatible with thickness of cell membranes. It's interesting fact. Well, and because DNA and RNA is chemically active substances, possible a lot of spontaneous random transformations. For example, it may be oxidation, in this case spontaneous oxidation. Cytosine in this case will be transformed to uracil. If it will be methylated cytosine, methyl groups, it sometimes signals for corresponding enzymes for gene expression, but it will be lead to transformation of methyl cytosine, which will be interact with guanine, to thymine. Spontaneous oxidation of methyl cytosine will be lead to forming of normal for DNA nucleotides, thymine. It again real, usual reason of mutations. Also can be transformation, oxidative domination of adenine spontaneously. Will be lead to hypoxanthine with properties of guanine, or guanine can transform it to xanthine. All of it lead to modification and usual reasons of mutations. And also possible other variants modifications. For example, any thousands in each cell of any thousands of purine bases can be spontaneously hydrolyzed from DNA structure and will be apurinic residues. Of course, it will be also reason for mutation and changing of genetic information. Our genome is constantly dynamic nucleotides and destroying of normal transfer of genetic information. For example, benzpyrene covalently associated with guanine, with amino groups of guanines, with forming of covalent derivatives. Or, for example, aflatoxins. Aflatoxins, toxins with, for example, appeared in decay apples. Decay apples lead to appearance of derivative of microbial synthesis, but did some paradoxal activity of cytochrome system, microsomal oxidation. Microsomal oxidation activity directed to detoxification by oxidation and forming of more soluble substances. But for aflatoxin, result of microsomal oxidation by cytochrome P450 lead to oxidized aflatoxin, and it can be lead to some not every time pleasure variants. For example, detoxificated dehydroxyaflatoxin derivative, non-toxic. Detoxificated complex with glutathione, non-toxic. But it can be also lead to covalent modification of nitrogen bases. Now again, guanine will lead to forming of covalent modification and mutations with destroying of function of genome reparation and cancerogenic effect. Next modification factor for DNAs is ultraviolet. Ultraviolet can be absorbed by pyrimidine rings. And if two pyrimidine rings appear together, for example, two thymines one to other, or thymine and cytosine, it can lead to forming of covalent bonds. In this case, it of course normal metabolism transfer of information from DNA will be impossible in this case. And each part of DNA must be excised and must be reparated. You can see multiple reasons lead to modification of genome by using of normal chemical properties of physical properties of nucleic acids. And but we have very effective systems for reparation of its modifications. Now, for example, base excision reparation, when we have modified nucleotide. In this case, it will be destroying of normal double-strand interactions, and it will be lead to excision of its part, and it may be more short or more long excision of part of one strand, and resynthesis of modified part without changing of genetic information. If it will take place in synthesis of second strand, so-called replication of DNA, in this case it will be stop of replication and reparation of excision of each part and reparated by using information of second strand and continuing of replication of DNA, but it will be lead to stop of normal metabolism of cells. But it's not such dangerous single-strand modifications. More serious mutations in double-strand damage, and reparation will be depend on stage of cell cycle. If it will be in period of cell cycle when in cells present double set of chromosomes, it will be in S phase or G2 phase. In this case, will be realized homologic variant of homologic recombination. When second chromosome, the same part of second chromosome will be example for reparation of double-strand break. In this case, it leads to the same reparation. Sequence of DNA will be non-changed. But if it will be take place in stage of cell cycle G1, when in cell absent corresponding homologic chromosome for each chromosome, in this case will be realized non-homologic end joining. In this case will be lead to ligation of broken after corresponding preparation of ends, ligation of double strand of DNA with missing of part of genome. It again real reasons of mutations and missing of any part of genome. You're going to see multiple effective dynamic events take place for DNA and RNA. And mutations and changing of activity of its enzymes, which will be lead to reparation of DNA. You can see correspondent enzymes which participate in this providing of reparation, will be lead to serious dysfunction of cells and development of carcinogenesis, and lead to development of correspondent symptom complex syndromes. I hope more detail will be studied it on the Department of Cancerogenesis will be studied. Onco department. Well, about packing of DNA. Stranded form of DNA lead to forming of near up to 2 meters of length strand. But you cannot see it, of course, its thickness is very low. But 2 meters of its DNA packed its nucleus, which you cannot see by eyes. How it possible to packet its structure with possibility of constant working with its sequence and transfer of its information. About it, we will tell some later. Так, dear students, now please some break. Near 7 minutes, please. Well, dear students, will be tell about packing of DNA. DNA in eukaryotes organized in nucleosomes. Nucleosomes, it structures which combine of special proteins, histones, and DNA. DNA winded on core of histone cores, and histone cores consist, no, again, you know, I hope, but I must repeat, four types of histones: H2A, H2B, H3, and H4. And two subunits of it, two molecules of it, each histone, lead to forming of nucleosome and winding of DNA on its structure. Now you're going to see some less than 2 winds, and totally on one nucleosome and linker path will be near 200 nitrogen bases, the bases pairs. And H1 histone also localized between nucleosomes, and it lead to packing of presence of H1 lead to more compact association of nucleosomes. Some about some details of structure. 8 histone molecules. And you can to see properties of histones. It near half mass of nucleus is provided by histones, very abundant structures, and consist a lot of positively charged amino acids. It lysine, arginine, histidine. Positively charged because DNA is rarely acid. Consists a lot of negatively charged, monotone repeating, negatively charged groups. And if we take DNA without proteins, it will be stake with impossibility of bending, and lead can be easily broken but not bent. But because the same charged, negatively charged, will be prevented. But interaction with positively charged of amino acid radicals will be lead to neutralization of negative charge and possibility of folding in three-dimensional space. Very conservative structures, and we have different islands of histone genes, and some copies identical, some different. And histones is not only structural elements, it also sensitive elements for modification of DNA. Any mutations in DNA will be lead to sensitive histones will be indicate place of mutation. It's not only structural but also sensitive mutation sensitive factors. Так, ну, and histones course and linking path depend on F1 histone. You can to see this molecular weight, number of amino acids, and number of lysine and arginine positively charged. Lead to next packing it so-called chromatin strand, double strand DNA, nucleosomes, chromatin structure, chromatin folded, chromatin stranded folded in next order of packing, at minimum 4 orders of packing lead to forming of chromosome. Next important factor for histone functions, and it lead to possibility of availability of genome, modification of histones, covalent modification of histones. It mainly or methylation or acetylation. Acetylated form will be lead to decompactization of genome. And acetylated or non-acetylated forms, you can to check is it part of genome will be expressed or not. It used for molecular engineering, for example. And binding of histones lead to possibility of gene transcription. For example, we can to see now internucleosomal region available for enzymes, transcription factors, or after binding with histones, it will be non-available. And transcription factors cannot achieve or can it histone state important for it. And it 3.2 billion nucleotides necessary to provide doubling when cell will be prepared to mitosis, to proliferation, to dividing. And it provided by providing of replication of DNA. Replication of DNA provided by dissociation of strands of DNA and synthesis of correspondent complementary strands. In prokaryotes, it takes place from one point, which will be directed on both sides, and prokaryotic DNA usually it's cyclic. Not every time, but very often. And it's possible relatively quickly. But for human and other mammalian genomes, it's impossible because replication will be for it necessary to use a lot of and will be provided by multiple replicational points. You can to see forming of so-called bubbles, replication fork, and for it necessary unwind of DNA and providing of DNA polymerase will be catalyzed. But it more complex. At first, replication is semi-discontinuous. Will be synthesized both strands, but one strand will be synthesized constantly, is so-called leading strand, and alternative strand will be synthesized by pieces, by fragments. It's so-called lagging strand. Continuous and discontinuous synthesis. And synthesis every time directed in from 5 to 3' end direction. Because nucleotides can be associated and added to a growing stream, growing polymer only on 3' end. Why? Because for synthesis of association next nucleotide, necessary nucleotide 3-phosphate. If it will be associated adenosine monophosphate, it must be adenosine 3-phosphate. Nucleotide 3-phosphate, adenosine 3-phosphate will be digested pyrophosphate, and digestion by pyrophosphatase will be lead to displacement of equilibrium to polymerizated form. And the source of energy will be nucleotide 3-phosphate. If it will be necessary added thymine monophosphate, thymine 3-phosphate. Again, cytosine 3-phosphate and guanylyl 3-phosphate. Set of nucleotide 3-phosphate. For synthesis of 3 billion nucleotides, or DNA from with 3 billion nucleotides, as correspond to 3 billion nucleotide 3-phosphates. Source of energy for elongation from 5' end is absent, and elongation limited by 3' end. The next problem. If will be only movement on one direction ahead of replicated system will be forming of coils and supercoils. It will be lead to destroying of possibility of replication. For avoiding of it, must be work special enzyme which will be unwinded DNA, double strand DNA, and winded after movement of its structure. And it unwinding enzymes called DNA topoisomerases. Topology part of mathematics we describe form of surfaces. And we have two types of topoisomerases, first and second type. Topoisomerases 1, first type, it nucleases really. It can to digest one strand and transfer of second its broken part across non-digested part with resynthesis of, again, resynthesis of rejoining or broken one strand. Second part, it digesting both strands, when both strands will be digested and rejoining. No, and in replication, first enzyme, it will be helicase. Helicase will be unwinded, but for preventing of reassociation of bonds and preventing of spontaneous hydrolysis of single-strand DNA, it very can be easily digested, spontaneously hydrolyzated. For avoiding of it, both strands will be filled associated with special proteins which will be associated with single strand associated binding proteins. And next, when we will have one strand of DNA, really possible synthesis of DNA complementary sequence, but DNA polymerase cannot bound with single strand. For it, really DNA polymerases it enzymes of reparation, which will be moved along DNA and check is both strands correct, have correct structure. And if will be empty place, result of excision by corresponding enzymes, it will be filled by DNA structures. But DNA polymerase cannot bound with single strand. Possibility of binding with single strand is RNA polymerases. And RNA polymerases will be bound with single strand DNA and will be lead to synthesis of short part of RNA, complementary RNA sequence, which called primer. And when will be appeared double strand DNA RNA, in this case DNA polymerase will be displaced RNA polymerase and bound with double strand, and will be moved ahead of DNA polymerase will be empty place and will be moved in direction from 5' end to 3' end. You can to see there. And alternatively, a second strand will be moved on opposite direction. And the fork will be go ahead, and one strand will be elongated by DNA polymerase more and more. It will be a leading strand, but for lagging strand will be appeared empty place. By filled, of course, single strand binding proteins, but it will be ready to binding of RNA polymerase. Again, RNA polymerase bound next primer. Next DNA polymerase will be elongate and will be achieve previous primer, and will be gradually move it ahead. You can see a leading strand will be moved ahead, but second will be synthesized by pieces, so-called Okazaki fragments. But will be appeared primers, and for excision of primers necessary next DNA polymerase, which called DNA polymerase 1, which will be excited, removed RNA primers, and DNA polymerase will be filled empty places. But DNA polymerase cannot create phosphoester bonds. It's provided by ligases, DNA ligases. And the result will be appearance of constant filled constant polynucleotide strand. Now you can see participate in this concert. It, of course, necessary DNA template, DNA helicase, which will be unwinded, single strand binding proteins, which will be prevent of reassociation and prevent of spontaneous hydrolysis. Primase RNA polymerase, DNA polymerase 3 main enzyme which for synthesis of DNA, DNA polymerase 1 for excision of primers, and DNA ligase, which will be fields of nicks of DNA. Now you can see DNA polymerase 3, it dimeric structure, which will be surrounded of DNA and move it along DNA structure, and it works simultaneously for both strands in the same direction. But leading strands go constantly, and it's not problem for it. But for lagging strands, it must be special loop structure for movement in the same direction. And it loop will be constantly changed. Size of its strand will be constantly changed, like trombone structure in music instrument. It's a called trombone-like model. You can see multiple functions which provide possibility of replication. And all of it moved with velocity some hundreds nucleotides per second for prokaryotes, some tens nucleotides per second for eukaryotes. And DNA polymerase work very effectively. It can to check not only nucleotide ahead will be bound with correspondent nucleotides, but can to check is backside of enzyme correct nucleotides will be present. If not, it will be reverse backside exonuclease activity and resynthesize correct nucleotide. But mistakes also every time present, and each cell dividing lead to accumulation of any errors, number of errors. And we haven't in our body two cells with the same DNAs. It every time any modification, some nucleotides from some nucleotides to some hundreds nucleotides in any case will be take place. Next, about RNAs. RNAs in cells. No, you know that usually students know that messenger RNAs, transfer RNAs, and ribosomal RNAs. But it's not only set of a limited set of RNAs. Also necessary to know about so-called small nuclear RNAs. It present in nucleus and provide possibility of excision of introns. We will tell about it some more detail. Small nuclear RNAs, it components of nuclear ribonucleoproteins, small nucleoproteins particles. It's important for excision of introns in pre-messenger RNAs. And very important regulative RNAs, so-called microRNAs, MIRNA. Short RNAs, double-stranded RNAs, which can provide regulation of gene expression. Really, it bound with the messenger RNA and provide its signal for digestion of its messenger RNAs. And now it's very effectively used for regulation of so-called knockout genes in regulation and modification of gene activity. For example, knockout mouse with knockoutated genes in mouse. For determination, is it gene important for function of living organisms, really? Well, next, how is it possible to study sequence of nucleotides? Sequence of nucleotides is the main factor for demonstration of possibilities and properties of living organisms. But for it necessary to have a sample of DNA. For it necessary extracted DNA. Now it's not modern system, but it also can be used for understanding of it. DNA, when you will have sample of DNA, you can or cloning of it in, for example, prokaryotic or yeast organisms for multiplication of it examples, corresponding gene, or maybe directly provide polymerase chain reaction for multiplication of it gene, and studying of sequence or maybe present of a gene in this living organism by more old it was gel electrophoresis or maybe micro capillary electrophoresis or sequence. All sequence of it events of it activity will be take place. Extraction of DNA, multiplication of it examples, and studying by correspondent methods. What is methods? One of it so-called, it was answered, but necessary to know it, but by historically. Not historically, but also now used for investigations of correspondent and the separation of correspondent genes. For example, you have sample of DNA. You can be digested by restriction enzymes, or can be broken by physically. Later, provide electrophoresis. Electrophoresis is method for separation by size, by separation of pieces of DNA by size. Later, transfer of separated parts of DNA to membranes, films. Membranes, it may be or capron films or polymer, any polymer membrane, or acetic acetylated derivatives. It's so-called blotting. Now you can to see for increasing of soaking of and blotting, provide paper towels. You have print blot of separated oligonucleotides in its film. And after this development by labeled probes, labeled at first it was irradiation, or now it by fluorescent probes. And you can to see where localize your at first present, or it is it present, and where it what properties of it part of DNA gene. You can to registrate mutations, and you can to registrate physical chemical properties. And later you can to accumulate it part of its gene and investigate it more effectively by sequence, for example. Now you can to see a real picture of electrophoresis in gel of agarose gel separation by size. When power supply, electrophoretic unit, and polymer of agarose for effective separation. Different multiple sample can be simultaneously investigated. And very effective method is polymerase chain reaction. Polymerase chain reaction is amplification, multiplication of correspondent part of genome. Previously, you must know what you can to study. And if you necessary to separate part of genome, you can to study. For it, previously, necessary to synthesize short part of complementary sequence, oligonucleotides. No sequence length of it oligonucleotides usually near 20. If you have, for example, 5, 10 nucleotides, this sequence may be present in genome multiplicity. But if you have near 20 up to 25 nucleotides, it will be possibility of individual, unique sequence. It will be much more. And it two nucleotides, which will be flanked, associated with size, both sides of interesting for you sequence, part of genome, will be bound with DNA. But previously, DNA must be separated. Because bonds between DNA, it hydrogen bonds, it weak bonds, non-covalent, increasing of temperature near up to 90 degrees will lead to full dissociation of both strands. Later, decreasing temperature and will be in presence of short oligonucleotides, which called primers. We talked about RNA primers, now it's DNA primers, which synthesized by chemically phosphoramidite method, for example. And primers will be bound with correspondent place of binding. Of course, second strand will be also try to bound, but molecular weight of primers less than a long chain second strand, and primers will be more preferentially bound with these sites. And later, synthesis of correspondent complementary sequence. And on each synthesized place will be a period place of binding for next for other primers. And after next dissociation of bonds and binding of next primers will be a period after next stage of polymerization will be a period polymer with molecular weight correspondent interesting path. And on each next step of polymerization, again dissociation, a melting binding of primers, and synthesis of complementary sequence. We will be accumulate complementary sequence of double strands with the same molecular weight. And it will be geometric progression. And after, for example, usual more than 20 cycles, we will be accumulated in multiple copy, which can be registered and checked by usual electrophoretic methods. Now you can to see check please thermal changing of modifications. Now it will be, for example, dissociation, denaturation, high temperature, melting, annealing, it binding of primers, and synthesis. Again, denaturation, annealing, binding of primers, and synthesis. And multiple time of events lead to accumulation of amplified product. Next problem is intermediate path between primers, real interesting path, and necessary to check. It different variants possible. One of ancient path is restriction by correspondent restrictases, which will be specifically recognized sequence with next electrophoresis. Or maybe determination by interaction with probes, fluorescent probes, with labeled by a fluorescent label. And the capillary electrophoresis can to determine size of its path. And direct sequence. Direct sequence, it's a lot of methods, but most effective was proposed and investigated by Frederick Sancher, also genial scientist, with double winner of double prize of you can do see it. He proposed used dideoxynucleotides. Binding of dideoxynucleotides with dideoxyribose impossible. And it will provide synthesis of DNA, complementary sequence, with addition of dideoxynucleotides corresponding test tubes. A sample of DNA must be separated on 4 test tubes. And in each test tube will be consist all set of nucleotides. You know that synthesis provided by nucleotide 3-phosphates. But now a little will be added, not fully, but a little addition of dideoxynucleotides. And when will be synthesis achieved this correspondent dideoxynucleotides, it will be lead to stop of synthesis. But other nucleotides will be continuing, and will be have set of derivatives with stop nucleotides on correspondent. Now, for example, adenine. Now it's stop on cytosine. Now stop of guanine and thymine. And you can do by after separation of by electrophoresis and labeling by for example, irradiated derivatives, or maybe now fluorescent derivatives of synthesis, you can do directly reading of sequence. Some hundreds nucleotides can be achieved. Now you can see Sanger with results of it. Sequence of nucleotides, sequence of genes with double Nobel Prize winner. And now it used by again for test tubes, or maybe in the same test tube with different labeling, different set of fluorescent labels. And electrophoresis by capillary electrophoresis, you can to see reading direct sequence of nucleotides. Now you can to see real situation in Sanger sequence. Now, for example, now it can be determined maternally inherited, used for forensic analysis, forensic science, human identification. But development of methods led to, for example, sequences. Sequences used because appearance as a result of synthesis of nucleotide sequence, it appearance of pyrophosphate. Using of pyrophosphatase can lead to possibility of flash of light when will be association of pyrophosphate and an enzyme which will be transformate energy to light. And you can do using of pyrophosphatase associated with correspondent binding of correspondent nucleotides, labeled nucleotides, you can do directly to determine sequence, by the way, sequence of nucleotides by registration of flash by association with correspondent wavelengths of fluorescent label. Next, it also can be used by microarrays, by fluorescence probes associated with different parts of sequence of nucleotides can be determined expressed and non-expressed groups of genes. For example, it can be used for determination of oncotransformation cells. What cells will be activated in correspondent genes. And so-called NGS sequences, it's a next generation sequence, when DNA broken on multiple short part of DNA and association with strands with correspondent synthesis of short complementary sequence for nucleotides associated with non-soluble resin, non-soluble support. And later, synthesis of complementary sequence with fluorophoric sequence with demonstration of sequence by correspondent color for different nucleotides labeled by different fluorophores, and it leads to direct sequencing of nucleotides. If it will be distributed on non-soluble, for example, glass support, you can to quickly registrate sequence of it short pieces. And next computer recombining, association of its sequences. But sometimes it may be difficult, because if you will have short part of limited part of relatively long sequences, it can be easy to get total picture. But if you have multiple short parts, and in our DNA a lot of repeating sequences, result of reconstruction of genome can be depend on fantasy of investigations. And next important development part of sequence, it using of direct demonstration by single strand DNA and pass across whole with size correspondent thickness of DNA. In this case, registration of electroconductivity can lead to, and the transfer of DNA strand across its whole, lead to constant changing of electroconductivity, and each nitrogen base will be influenced individually. And you can to see sequence of nucleotides by sequence of electroconductivity. Yes, now it very effectively developed methods. And the advance of whole genome sequence lead to next problem, ethical problem, because information about genome individual patients, about himself, yourself, can used against you, and so-called personal genetic data. And it appeared new. You can to buy information about your genome. It near one up to $1,000, sometimes less, up to $100 maybe. But in future, but your information at first problem, you cannot to effectively to describe its information. And second, its information tell about your individual genetic properties. Well, now we'll be finished our lecture. Thank you very much for your attention. Until the next lecture, we will continue. Goodbye.